This video introduces the basic concepts of nucleic acid transfer, as well as some of the chemistry and cell biology underlying transfection technology. The scientists at MIRIS are truly transfection experts. Our team is based in Madison, Wisconsin, and has been focused on gene delivery technology since 1995. In addition to technologies for transfection, MIRIS has made advances in electroporation, as well as for labeling and visualizing nucleic acids such as DNA and RNA. Today's talk is divided into 3 sections. First, we will discuss the basic biology of transfection. Next, we go over the process of performing a transfection and some of the key parameters that define experimental success. Finally, we go over some of the primary transfection applications in which transfections are used. Let's begin with the biology of transfection. Transfection is the process of transferring nucleic acids into cells. Examples of naturally occurring nucleic acid transfer include the fusion of gametes in animal reproduction, infection of cells by viruses, the movement of DNA molecules called plasmids from one bacterial cell to another through a process called conjugation, as well as plant reproduction. In contrast to these naturally occurring processes, transfections are performed by scientists to deliver DNA to cells growing in the laboratory. But the technology used to deliver DNA draws on inspiration from other areas of biology. The term transfection primarily describes the introduction of an exogenous nucleic acid into tissue culture cells. For example, this could be circular plasmid DNA or messenger RNA for gene expression, or siRNA for gene silencing. This slide illustrates what happens in a typical DNA transfection. The 3 images show Cas7 cells that have been transfected with DNA, which is stained blue. The DNA that was delivered to these cells encodes a yellow fluorescent protein. As the DNA gets transcribed into RNA, and the RNA gets translated into protein, you begin to see the yellow fluorescence accumulating at the 8 and 20-hour time points. In contrast to DNA transfections where you are expressing a gene, siRNA transfection can be used to inhibit gene expression. In the image shown in A, HeLa cells have been transfected with an siRNA that has been fluorescently labeled, which you can see as green spots. The nuclei have been stained blue, and the edges of the cell are stained red. In B, you can see the level of gene knockdown caused by the transfection of this siRNA. The level of expression is normalized to 100% for cells that were only treated with a transfection reagent. For cells that were treated with a transfection reagent and siRNA, the level of expression falls below 20%. Regardless if the siRNA was unlabeled or labeled with a fluorophore such as Cy3, Cy5, fluorescein, or rhodamine. There are a number of different ways that cells can be transfected. 2 broad categories of transfection approaches are chemical and mechanical methods. Chemical approaches include the use of calcium phosphate, lipids, polymers, and other chemicals. Mechanical approaches include electroporation, directly injecting cells with DNA, biolistic delivery or the use of a gene gun, as well as methods employing magnetic fields. You could be wondering why transfection reagents are needed in the first place. The reason we need transfection reagents is because if you simply add nucleic acids to tissue culture cells, nothing happens. This is because mammalian cell membranes are inherently negatively charged, and this causes a repulsion between the negatively charged DNA you intend to transfect and the negatively charged plasma membrane. So in this process, the role of the reagents, which are typically made up of positively charged chemicals, is to neutralize the negative charge on the DNA. The resulting combination of reagent and DNA is referred to as a transfection complex. This complex has a net positive charge which enables it to be taken up by cells. Now imagine a typical transfection, where a cell is growing in tissue culture medium surrounded by transfection complexes. Some of the positively charged complexes will bind to the negatively charged cell membrane and get taken up into the cell by a process called endocytosis, then move out of the endosome into the cytoplasm. For DNA transfections, the DNA must also enter the nucleus to be transcribed into RNA. The use of chemical transfection reagents began with calcium phosphate, which utilized calcium ions to neutralize the charge on DNA. More modern transfection reagents often use combinations of lipids and polymers which combine with DNA to form what is called a lipopolyplex. The blending of these different compounds can have synergistic effects that provide higher transfection performance. This slide summarizes the process of transfection. As we discussed, transfection refers to the delivery of exogenous nucleic acids to cells. The nucleic acid could be plasmid DNA, messenger RNA, small RNAs such as siRNA and microRNA, or other types of nucleic acids. The first step in the process is the mixture of the nucleic acid to be transfected with a transfection reagent to form a transfection complex. Second, the positively charged complex associates with the negatively charged plasma membrane. Third, the complex is taken up by the cell through the process of endocytosis. Next, the nucleic acid must escape from the endosome and enter the cytoplasm. In the case of most RNA transfections, this is where the journey ends because the cytoplasm is the site of action for gene silencing or mRNA translation. For transfections where nuclear localization is necessary, such as with plasmid DNA, the nucleic acid must also cross the nuclear envelope, which typically occurs during cell division. We will now move on to discuss the steps involved in performing transfections and the key parameters that can be optimized in transfection experiments. Performing a transfection using a chemical transfection reagent is very simple. The transfection reagent and nucleic acid, such as plasmid DNA, are mixed together in serum-free medium and incubated at room temperature for 15 to 30 minutes to allow transfection complexes to form. Following this short incubation, the transfection complexes are added to tissue culture cells growing in complete medium. One of the major factors impacting transfection efficiency is the composition of the serum-free medium used for complex formation. Serum is avoided at this step because it contains nucleases that can degrade the nucleic acid before it has had time to associate with the transfection reagent. Some antibiotics, such as kanamycin, are cationic and will inhibit transfection complex formation and should be excluded from the complex formation step. Similarly, some serum-free media contain polyanions, such as heparin or polydextran sulfate, that may inhibit formation of active transfection complexes. Certain transfection applications require surfactants, such as Embryonic, for a long-term growth and increased protein yields. However, these compounds can inhibit transfections at high concentrations and should be avoided altogether during complex formation. The next critical component of the transfection workflow is the nucleic acid. Nucleic acids used for transfection should be highly pure and free of endotoxin. Ideally, DNA used for transfection should have a 260 to 280 ratio of 1.8. Endotoxin is a lipopolysaccharide present in the outer membrane of gram-negative bacteria such as E. coli. Many maxi-prep kits will afford the desired level of purity, but it is important to verify that your chosen plasmid prep kit states that it yields transfection-grade or transfection-ready DNA. The importance of avoiding endotoxin is illustrated in this bar graph. This data shows COS7 cells transfected with a luciferase plasmid. When you use a very pure, commercial-grade plasmid, you achieve high levels of expression compared to a crude plasmid prep containing high levels of endotoxin. If you take this same crude plasmid prep and remove the endotoxin using our MiraClean endotoxin removal kit, you can restore high expression levels. The amount of time that you allow the complexes to form is also highly important. Most of our transfection protocols call for an incubation time of 15 to 30 minutes. During this time, the reagent is coating and condensing the nucleic acid, and the particles are growing in size. Here's an example of complex formation time compared to luciferase activity in CHO-K1 cells. Before the 15-minute time point, the complexes are too small for this cell type, and beyond the 30 minutes, the complexes are likely too large, and you start to see the efficiency begin to drop off. This is one of the main reasons that you want to prepare your transfection complexes fresh with every experiment. Cell culture conditions such as confluency also play a role in transfection efficiency. Here is an image of cells showing confluencies anywhere from 25% confluency to 90% confluency. Performing transfections on the very low end of confluency can lead to problems with cell health, whereas performing transfections on completely adherent cells will give low efficiency. To illustrate this point, here's a graph showing 3T3 cells transfected at 75% confluency and 100% confluency. The cells at 75% confluency show very high efficiencies, whereas the fully confluent cells, which are likely not dividing, do not transfect well. In order to ensure success in your transfection experiments, you should pick the optimal harvest time, or when you actually do your downstream experiment following transfection. The optimal harvest time is primarily determined by the goal of your experiment. For example, typical harvesting times for overexpression studies are between 24 and 72 hours. For gene knockdown, it is generally recommended to wait slightly longer, between 48 to 72 hours. Of course, this depends on the stability of your target mRNA, and if you're assessing knockdown at the protein level, the half-life of the protein plays a role as well. mRNA, on the other hand, is very quick. Expression can be seen as early as 4 hours following transfection and usually peaks prior to 24 hours. For specialized applications, such as therapeutic protein production, a longer time frame may be desirable, anywhere from 2 days to 2 weeks, to get the appropriate cellular biomass and associated protein yield. For an example of this effect, here are suspension CHO cells harvested at different time points using several different reagents, and you can see in each case that there is an increase in the overall expression at longer incubation times. One last factor that we will go over is reagent formulation. This image panel illustrates the time-dependent and formulation-dependent effects on cell health using different reagents. These are HeLa cells that have been transfected with GFP-encoding plasmid DNA using several different reagents. On the left are two Mirus reagents, Transit LT1 and Transit 2020, and in comparison is a popularly used competitor reagent, lipofectamine 2000. Each of the reagents have distinct formulations. At the 8-hour time point, you really do not notice any differences in cell morphology with any of the reagents. However, as you progress to the 24-hour time point post-transfection, the lipofectamine formulation causes substantial cytotoxicity. The cells are shriveled up and rounded and have detached from the plate compared to the healthy and adherent cells that are transfected with the transit formulations. This also correlates with the GFP efficiency. As you can see in the two reagents on the left, because of the higher cell viability, you are left with significantly more GFP-expressing cells. So in addition to optimizing parameters pertaining to complex formation, cell culture conditions, and harvest timing, it is highly important to choose a transfection reagent formulation that is compatible with your cell type. In this last part of the talk, we will explore a few different ways that transfection is used in life science research. There are many ways that researchers utilize transfection. Only a few are mentioned here. In the material that has been presented so far, we have already touched on the use of transfection for basic applications such as gene expression and gene knockdown. Two additional common applications are stem cell reprogramming and genome editing. Due to the risks associated with viral gene delivery methods, stem cell researchers often utilize chemical transfection for gene expression, RNAi, and reprogramming experiments. Transfection impacts multiple steps in this workflow. For example, in reprogramming experiments, a combination of transcription factors called Yamanaka or Thompson factors can be transfected into adult somatic cells to generate induced pluripotent stem cells, or iPS cells. These iPS cells can be subsequently transfected with plasmids encoding differentiation factors, or drug-selectable cell-specific markers, to generate a range of defined cell types such as neurons, adipocytes, and cardiomyocytes. Lastly, transfection can be used to deliver plasmids, mRNA, or siRNAs and microRNAs to these differentiated cell types. Cell lines that are commonly used for reprogramming, such as MRC5 and BJ fibroblasts, can be efficiently transfected with minimal toxicity to deliver mRNA using the Mirus transit mRNA transfection kit to generate iPS cells. Through a collaborative work with the stem cell technology company Cellular Dynamics, we have observed high-efficiency transfection of human iPS cells with plasmid DNA. We have also observed efficient transfection of iPS cell-derived cardiomyocytes. Importantly, not only reprogrammed stem cells, but stem cells isolated from embryos can be transfected, as well as we have seen in mouse embryonic stem cells using our Transit LT1 transfection reagent. Another popular transfection application in biology research is genome editing. The example we will discuss today is using transfection to correct a disease gene in the genome of a cell. For instance, say you have a cell which has become cancerous due to a particular mutation. One of the ways you can correct mutations in genes that lead to disease is to transfect DNA that encodes an enzyme that works like a pair of molecular scissors. Once the molecular scissors enter the cell, they can cleave the disease gene and restore the cell to its normal healthy state. This concludes our discussion of transfection technology. If you would like to know more details about nucleic acid delivery, please visit our outreach page at www.mirusbio.com/outreach.